Good afternoon to you all. I am Dr. Mrs. Madhuramozhi Govindarajalu, Head of the Department and Associate Professor in Zoology, A.D.M College for Women, Nagapattinam. Today I am going to take you the linkage and crossing over. This is for the first MSc students, and this linkage, before going to detail about the linkage, all of you should know the structure of the gene and the position of the gene. Where the genes are located? The genes are located in the sex chromosome as well as in the autosome. So generally it is seen in the chromosome. So the process of linkage as well as the crossing over takes place during gametogenesis, because the genes present on the homologous part of the sex chromosome, they exhibit the phenomenon of incomplete linkage. So the linkage may occur in the autosome, that is in the body chromosome, and this may also occur in the sex chromosome. So if the linkage is seen in the sex chromosome, these chromosomes, these genes are said to be the sex linked genes. So linkage groups, this was first detected in fish by Winger during the year 1923. So the example for the genes which are seen as the linked genes in the chromosome is the 6th chromosome, that is 16th gene, as well as XEL gene. This can be seen in guppy. This is the X chromosome, and although both genes may cross over to the Y chromosome also. So before going into detail about the X and Y chromosome, all of you should know the sex chromosome, because in human there are 23 pairs of chromosomes, and among these 22, first 22 pairs are said to be the autosome or the body chromosome, and the last chromosome is the sex chromosome, because in female 2 X chromosomes and in male X and Y chromosome. So those chromosomes which are linked in the X chromosome are said to be they are responsible for the X linked character. Likewise in man, those chromosomes, that is the genes linked in the Y chromosome, they are said to be the Y linked genes. So totally human being having more than 23,000 to 25,000 genes, and they are in close number to that of other mammals and other animals like mice, roundworms, small flowering plants including the Arabidopsis species, Thalina, and also the fruit flies. For example, the Drosophila melanogaster we are taking for the various genetic studies, this also having more than 14,000 genes. Yeast has 6,000 genes. E. coli bacteria having 3,200 genes. And also among the fish, the zebra fish, this is very important in genetic aspects. They are having more than 5,000 genes. So a single chromosome bears several genes. You already know the genes are arranged in a linear manner, that is in a beaded structure in the chromosome, because the chromosomes you can see inside the nucleus, isn't it? So after the genetic information are transcribed, they are coming through the nuclear pore to the region of protein synthesis, that is to the ribosome. So here two important functions which are carried by the gene. The first one is the carrying of genetic information, and another function, this is the dedicating the dictation of the species to what type of protein that our body require. This will be determined by the gene. So two important functions, one is the genetic information carrier, another one is the protein synthesis. These two processes are completely depending on the genes. So the genes present at the same locus, they have a tendency to link together. This is the linkage. So linkage is defined as the tendency of the genes to remain together during the process of inheritance, that is during the process of gametogenesis. They are going to the next generation. So when two loci were linked genetically on the same chromosome, they did not segregate, and this is the extent of the linkage, or closer the genes, stronger the linkage, and vice versa. So linkage of genes is in the linear fashion in the chromosome, and also we can see the show the linkage are located on the same chromosome, and a linkage group is formed by all the linked genes in the chromosome. So the strength of the linkage between the two genes is directly proportional to the distance between them. That means if the genes are arranged closer, the linkage will be closer, isn't it? The tight linkage. In some time there may be the possibility of the linkage, incomplete linkage also occurs. So complete linkage means when two or more genes which tend to remain together on the same chromosome and are inherited together for many generations. So this phenomena is called complete linkage. This also occurs, but sometimes when chromosomes do not undergo any breakage by accident during gametogenesis. And incomplete linkage means when the linked genes tend to separate on some occasion during the course of inheritance. That is the process of crossing over. At the time the incomplete linkage occurs. So this is the incomplete linkage is nothing but the process of crossing over during gametogenesis. So what is gametogenesis? Development of the gametes. This is taking place only in the sex chromosome, isn't it? So the genes present in the homologous part of the sex chromosome, they only exhibit the phenomenon of incomplete linkage. So chromosomes are linked group, they contain the group of genes, they are the linked together, but during incomplete linkage, that is during crossing over at the time of gametogenesis, because during in the crossing over occurs during the Pachytene stage. Where occurs the Pachytene stage? When you take the cell division, mitosis, meiosis, and mitosis is taking place in the somatic cell, meiotic division in the reproductive cell. So meiotic division includes two stages, one is meiosis 1, meiosis 2. So during the meiotic division, particularly in the prophase stage, which includes various substages like leptotene, zygotene, diplotene, pachytene, and diakinases. So during the Pachytene stage only the crossing over that is occurs. So chromosomes are the number of linkage group is the number of types of chromosome of the species. So for example, in human 22 autosome, I told you already, no. So 22 autosomal linkage group, and X chromosome the linkage group is group, and Y chromosome also we can see the linkage group. So the first direct evidence of the linkage, they came from the studies of Thomas Hunt Morgan. He only investigated several traits which are followed on the X-linked pattern of inheritance. So below the figures they are illustrating the experiment. So three traits of body color, this is taken in the Drosophila. One is body color, three trait means three characters. So body color, eye color, and wing color. So after we can see the first generation with the body color, wing color, and eye color, and the Y, the male, which is having comparatively different red eye color, black body, and the wing is vestigial, somewhat little. But when you take the female, yellow body and yellow, that is the transparent color in the eye, and with the long wing. So F1 generation, there is the linkage of the character occurs. So entirely the eye, which is in the F1 generation, they are having red eye color. So what are the dominant characters? This we can find in the F1 generation. So according to Mendel's law, this is proved, isn't it? So there are a mixing of other pattern also, that is instead of only the same grey body, we can also see the yellow body and white eye, miniature wing. Miniature means small wing. Grey body, red eye, normal wing. Grey body, red eye, miniature wing. So we can see multiple variations in the F2 generations also. So Morgan observed much higher proportion of the combinations of the traits which is found in the parental generation. So according to Morgan's explanation, all three genes are located on the X chromosome, that is in the sex chromosome. So similar to human, this Drosophila is also having the X chromosome, X and 2X chromosome in the female and the X and Y chromosome in the male. So therefore they tend to be transmitted together as a unit. But here we can see the yellow body, white eye, miniature wing in large quantity, the grey body, that is the parental generation, which will be in greater amount, both parental number, but we can also see the intermingled species also. So Morgan observed, Morgan's hypothesis was that this eye color gene is located on the X chromosome. So here the males are hemizygous because there is no homologous gene on the Y chromosome. The original mutant male's genotype was the small w, capital Y, that is hemizygous with a recessive allele. Females may be homozygous or heterozygous because the wild type female in the original cross was the homozygous for red eye. The F1 flies were the females which are said to be small w, that is the small vestigial wings, and the females are heterozygous, males hemizygous dominant. Likewise the F2 data also complete crisscross inheritance. What is crisscross inheritance? That is the character which is imprinted in the X chromosome. This will be transmitted to the next generation, particularly to the male. But the males are acting as a carrier, they won't express, and it will again express only in the next generation. So through the daughters are said to be the carrier of the disease. For example, we can see the sex-linked character, that is night blindness and hemophilia. So the father may be having that is the grandfather will be having the disease, but this will be transmitted to his daughter who is heterozygous, and this disease will be transmitted to her son. Is it clear? So male to male through the daughter. So this is called the X-linked or sex-linked or crisscross inheritance. Morgan's hypothesis was confirmed by an experiment reciprocal to the original cross. A white-eyed female was crossed with the wild type male. The results are the reciprocal cross. All the F1 female had red-eyed and all F1 males had the white-eyed. So these F1 results are different from those in the original cross. So all the F1 had red eye, and the F1 from the reciprocal cross interbred, they are said to be F2. So this is the discovery of the X-linked inheritance. He showed that when results of reciprocal crosses are different, the ratio differ between the progeny of the different sexes, and the gene involved is likely to be sex-linked or X-linked genes. So this was strong evidence that genes are located on chromosomes. So we already told about the arrangement of the genes. The genes are arranged in a linear manner inside the chromosome. Morgan also received 1933 the Nobel Prize for Physiology or Medicine for his work and the appearance of rare phenotype. This is also associated with the inheritance of a particular chromosome. So sex-linked genes are located on the X chromosome. The wild type eye color of Drosophila is dull red but pure line, which white eyes are available. The alleles for the white eye is recessive. This is said to be recessive. And white-eyed female, they would possess 2 X chromosome and 1 Y, and that red males, they are possessing the single X chromosomes. So this is the red-green color blindness, which is inherited as an sex-linked recessive trait in human. I already told you, a sex-linked, that is color blindness as well as hemophilia. These are the disease. But Y-linked means this will be seen in the even in the next generation itself. If a father is having the Y-linked disease like hypertrichosis, long hair in the ear, this will be transmitted to his son immediately in the next generation. This is also called Y-linked character. But his sex-linked or X-linked will be seen in the crisscross inheritance. Not in the next generation, it will be explained in the next, that is the second generation. So, from the grandfather to daughter, then daughter to grandson. So, it is the crisscross. So, human eye, they detect only 3 colors. This is the explanation, red, green and blue, but affected woman possess X-linked recessive trait to her son, but not to her daughter. So, affected man possesses the trait to his grandson through his daughter, but never to his son. Because that particular X chromosome, no, this is the recessive chromosome, even though it goes to the daughter, but it cannot be expressed because of the dominant X she is having, isn't it? So, it comes to the next generation to his grandson, at the time only it can be expressed. So, the pattern of inheritance exhibited by X-linked recessive character is sometimes called as crisscross. So, 4 general pattern of inheritance are observed. One is the autosomal recessive. So, the linked disease are cystic fibrosis, and it appears in both male and female children of unaffected parents, and autosomal dominant, it is the Huntington disease, and the affected males and females appear in each generation of the pedigree. Affected parent transmit the phenotype to both male and female children. Then X-linked recessive, this causes the disease hemophilia. So, many more males than the female, they show the disorder. All daughters of an affected males which are said to be carrier. So, none of the sons of the affected male, they won't show any disorder. Only the females are getting the sex, that is X chromosome, but they are said to be carrier. So, it is the X-linked dominant, and affected male pass the disorders to all the daughters, but to none of their son. So, affected heterozygous female, they married to unaffected male, they pass the condition to half of their son and the daughters. So, this is the explanation for the pedigree. So, after the disease is introduced into the family, in generation 2, it appears in every generation, and they show the dominancy. Fathers do not transmit the phenotype to their son. So, it is seen. The further apart 2 genes on the same chromosome, the more it is likely that a recombination between them will occur. So, 2 genes are linked if the recombination fraction between them is small. So, linkage map estimate the distance between the gene. The closer together 2 genes are, the more likely they will be inherited. If the genes are arranged in a closer manner, they will be inherited in a strong manner. So, crossover frequency are related to distance between the genes. So, linkage map show the relative location of the gene. Morgan found that linked traits are on the same chromosome. So, chromosomes, the not genes, they assort independently during meiosis. Assort means they group. So, linked genes are not inherited together every time. So, chromosome exchange homologous genes during the meiosis. So, there is crossing over takes place during the meiotic division, particularly in the packaging stage. So, the chromosomal materials are exchanged between the non-sister homologous chromosomes. So, genes that which are far apart on the same chromosome, they may independently assort from each other. This is due to crossing over. So, in diploid eukaryotic species, linkage can be altered during meiosis as a result of crossing over. That means, if crossing over occurs, the linked genes will be not as such, it will be crossover, or there is mixing of the gene. So, crossing over occurs during prophase 1 of meiosis at the bivalent stage. The non-sister chromatid of homologous chromosome they exchange the genic material, that is the DNA segments. So the physical exchange of parts of non-sister chromatids of homologous chromosomes following synapses at meiosis, this is known as crossing over. So when you comes to the frequency of crossing over of a given pair of gene is not constant, it is a part of meiotic mechanism during gametogenesis. So this brings about the variation, and this leads to evolution through natural selection, because variation is very, very important. Without variation, without mutation, no variation. So without variation, there won't be any species, new species, or there won't be any origin of new species. Here the natural selection is having an important role to check the species in a natural level. So coming to the crossing over here, there may be the single crossing over or multiple or double crossing over which may occur. So only one chromatid of each chromosome is involved, means this is called single crossing over. So both the chiasmata may between the same chromatid or between different chromatid. So all the 3 or 4 chromatid crossing over occur, means this is involved in the process of double crossing over. So see the arrangement of the gene, that is the EFGHI, but after crossing over what happens? The EFGHI, instead of this the H and I will come here. So this will be the centromere, the chromatid, the double crossing over. That means in 2 places the crossing overs occurs between the non-sister homologous chromosome. So multiple crossing over means there are more than 2 chiasmata. Chiasmata means crossing, that is the point where the junction or the fusion or the exchange of the genic material is taking place. So corresponding to the number of chiasmata formed, it is called triple chiasmata or quadruple. If the 4 places crossing over occur or 4 chiasmata occurs, it is called quadruple, and so on. So in diploid eukaryotic species, linkage can be altered during the meiosis as a result of crossing over. So crossing over occurs during first meiotic division. And what are the significance of crossing over? So this provides a direct evidence of the linear arrangement of the gene in the chromosome, and we can easily construct the chromosome map. And the crossing over gives rise to a new combination of the gene, and hence the variations in offsprings occur. So diploid cells and the stage where the crossing over. So without crossing over, the linked allele will be segregated together. So the haploid cells having the same combination if there is no crossing over. So arrangement of linked alleles, they have not been altered if there is no crossing over. So crossing over can reassort the linked alleles. So these haploid cells having the combination of the alleles, they not found in the original because of the crossing over. We can get new variety. These are termed parental or non-recombinant cells. So this is the new combination of the allele. This is the result of genetic recombination, and these are termed non-parental. So Batson and Punnett discovered 2 traits that did not assort independently. So they suggested the transmission of the 2 traits from the parent. 2 trait means 2 character from the parent was somehow coupled. So the 2 traits are not easily assorted in an independent manner. However, they did not realize that the coupling is due to the linkage of the 2 genes on the same chromosomes. Batson and Punnett experimented. They discovered 2 traits that did not assort independently, and this is a hybrid dihybrid cross that is expected to yield 9:3:3:1. But we can see during the interaction of genes, we cannot get this 9:3:3:1. We are getting various different ratio, like the duplicating gene and suppressor gene, and they are having various different types. So crossing over, that is the variation, may occur between non-sister chromatids at the chiasmata. So the segment of the non-sister chromatid may break and they reattach to other chromatid, and the chiasmata, that is the chiasma, are the site of crossing over. This is the particular place where the two non-sister chromatids of the homologous chromosome touch and they make a cross, that is the chiasmata. Thank you.